last night, so I gotta
Sorry.
click. Yeah, no problem.
There.
So we had our first snowfall last night, but that doesn't change the topic of what I'm going to speak about. What I want to present to you is the updated soil food web. This is uh, uh, a a talk that basically goes through the science behind organic gardening, and at the end of an hour, you're going to know everything you need to know to be a fabulous organic gardener and reasons why you would not want to be a chemical gardener. But in any case, I call it the updated soil food web. Um, let's see if I can get this to work. With all the practice we had in COVID, I guess I got to put play. Yeah, that might make some sense. All right, there we go. Now I have to do an adjustment. It's crazy. I don't know if you can see that. I am the author of. Let me just get this. I'm sorry about this, folks. Again, with all the practice we had during COVID, you'd think once it's over, we'd get it right. Okay, so I am the author of the Teaming with Microbes series, and as a result, I had a trilogy, and I called myself Lord of the Roots because I thought that was kind of funny. And then these are the books in the trilogy. Gee, it's doing it again. I can fix that. Sorry, folks. So the trilogy, it turns out, was not enough because we did have COVID and I had nothing else to do. So I wrote a fourth book, and now I have a quadrilogy. And the books basically deal with the fact that plants produce exudates, things that drip out of the root system by using their photosynthetic energy and by putting together the nutrients that it obtains from the soil. And these are the exudates, a great picture of the exudates. And these exudates attract microbes. And these microbes are what run our gardening system. Now, I could talk about it using this slide, but it's a little ununderstandable. So let me go this way, make it a little bit more understandable. About 50% or so of the photosynthetic energy of a plant is used to produce things that drip out of the roots. You're sweating. Your sweat is the exact same thing. It's an exudate. These exudates drip out of the roots, and they're designed by the plant to attract bacteria and fungi, because the bacteria and fungi don't produce their own carbon. They love these exudates because these exudates are full of carbon. So they're right there next to the roots in what's known as the rhizosphere, and they're eating away, having a great time. And then along comes their predators, nematodes and protozoa. And the nematodes and protozoa eat the fungi and the bacteria because they also need carbon. And there is carbon contained now in those fungi and bacteria as a result of eating the exudates. And the nematodes and protozoa devour these fungi and bacteria, and then they excrete the excess that they don't need. And what they excrete turns out to be plant usable nutrients. They break down the fungi and bacteria into nutrients that are usable by the plant. They actually have an electrical charge put on them so that they can go into the plant. And that's basically the soil food web that we learned in 2006. It was popularized by a woman named Dr. Elaine Ingham, but even that's a little too confusing. So let me show it this way. And then of course you've got the little guys, the nematodes and the protozoa get eaten by bigger guys all the way on up the chain. So let's look at it this way. There's a bunch of trees. It's a Friday afternoon. This is Fox Island. And these trees are sitting around talking to each other through the World Wide Web. And one of them says, let's have some Japanese food tonight. And a littler one says, nah, we did that last week. Let's have some Mexican. And the third one says, no, let's have French. But the big tree wins. And so they're going to have Japanese food. These are the roots of those trees. Let me put them in root position. And all that they have to do in order to get the Japanese food is produce an exudate that they drip out into the soil right there in the rhizosphere, and they attract the Japanese fungi and bacteria that they are going for. Now, if one of the plants had said American food and they decided to have American food, they would change the exudate. The plants are capable of changing the exudates and dripping them out and attracting American food. So they can change the exudates in order to obtain nutrients and results that they need. This is an amazing thing, because we look at plants, they can't walk, they can't run away, they can't gather things, but they can produce these exudates, which do all sorts of things for the plant right there in the soil. So this is the soil food web that we learned back in the very early days. This is the basis of Teaming with Microbes, my first book, which has been translated now into 10 languages, not because it's a great book, but because this is how we learned that the plant feeds itself, not through Miracle Grow, not through Peters, but through their own natural system. And it got the name microbe-mediated solubilization of soil nutrients, 2006. So this is teaming with bacteria. And it's, again, a very simple system. The little guys get eaten by the bigger guys, and there's all sorts of consumption and decay, and you end up getting nutrients which go into the soil and go into the plants. It's a very simple system. All right, so then several years later, it became very clear that we needed to amend that view because there was something else going on involving those fungi. So it was called mycorrhizal fungi symbiosis. Now, you remember those bacteria and those fungi that are attracted by those exudates. Well, some of the fungi don't get eaten. Instead, they get called by the plant. The plant sends out a signal, and they enter into the plant, and they move between the cells of the plant, and they transfer nutrients, which they obtain from the soil, into the plant in return for the exudates that they get. So this is a picture of mycorrhizal fungi entering into a root and getting into the insides. And it's just a fascinating thing. And it turns out that about 90% of plants form this symbiotic relationship. And we've known about it since the 1890s, but we were never able to really duplicate the fungi. And so it had no commercial application. And if there's no commercial application, scientific research often slows down to a standstill. This is a picture of one of the 7 different kinds. This is an important kind that associates with your trees. And around all your tree roots are these hyphal sheets, these fungal hyphal sheets. And then some of these fungal go inside, and they form structures in between the plant root cells. And they're very elegant. Now, they've been very studied. There are some 300 or so different kinds. We're capable of making 8 to 16 of them. I can't remember what the exact number is, but they sleep around. And so one particular type of mycorrhizal fungi will infect, and that's the right word, many different kinds of plants. This is the kind that does the trees. And then this is sort of a depiction of what is really going on. You have all of these wonderful small mycorrhizal fungi that are able to go out into the soil in an area where the plant root itself can't reach because it's too big. And they collect phosphorus and water. These are the things that they bring back, and they bring it back to the plant. And the plant says, thank you very much. Here's some exudate. Go back and get me more. And they have this wonderful symbiotic relationship. And it's a good thing they do, because, again, the root itself goes into the soil, but it forms a depletion zone. And so by having these mycorrhizal fungi go out into the soil, it extends the reach of the root itself. which is an important thing, obviously, because you don't want plants growing in the depletion zone, because their only other choice is to just simply grow down deeper and leaving all these great nutrients there. All right, so this is how a mycorrhizal fungi works if you don't have a telescope to watch it. So you've got a bunch of seedlings, two of them sitting out in the sun, again, Fox Island, and one of them says, gee, I would really like a bologna sandwich. And so what does it do? Well, it mixes up the right exudates. It puts those exudates out into the soil, and it attracts the kind of fungi that it needs to make a bologna sandwich, which it does, and it delivers it to the seedling. The seedling now has established this relationship. The relationship continues, and the seedling grows as a result of the work that the mycorrhizal fungi is doing in return for getting those exudates. So this is a very important addition to the basic 2006 soil food web picture, and these mycorrhizal fungi do some really fun things. They can associate with an individual plant, or they can associate with more than one plant and form that web. It's not quite the worldwide web that's been described in the news, but you do get sharing between individual mycorrhizal fungi and different plants. All right, so that was about 2010. That was called mycorrhizal fungal symbiosis. And then we started to learn about something known as endophytic bacterial symbiosis. And this is the basis of my fourth book. This would be the second book I would read, incidentally, after Teaming with Microbes, if you were reading the series, because it's such an important new addition to the way the soil food web works. So let's get into that. It's called endophytic, meaning inside, endophytic plant, inside the plant, bacterial symbiosis. And it features something known as rhizophagy, root eating. A discovery that was made back in 2010 in Australia by a bunch of scientists working at the university, Southern University, I think it was. And what they discovered was that there were bacteria just inside plant roots, which they thought occurred as a result of plant root cells engulfing these bacteria. And they called that rhizophagy. And unfortunately, they called it rhizophagy or something of that sort because they had an Australian accent, but I call it rhizophagy. And unfortunately, they ran out of funding. And so the whole concept just sort of languished until 2018, when scientists at Rutgers University picked up this discovery and using new kinds of microscopes really figured out what was going on. And it is a whopper. So let's go into it. So again, some of those fungi don't get eaten. They form the mycorrhizal fungus that are attracted by the exudates. And some of those bacteria, they also don't get eaten. They form what's known as the rhizophagy cycle. So I want to first point out that bacteria, incredibly small. This is the proverbial pin, and there are the bacteria on the tip of the pin. So they're incredibly small. They're everywhere. They multiply every 20 minutes. They're fascinating. And we don't really study enough. We study dinosaurs. We don't study things like microbes that are important. But these are really incredible. So I want you to keep this picture in mind as we go through some of these next slides. So the bacteria are attracted by that exudate, and here are some bacteria, bunches of bacteria that are attracted to the root tip of a plant, and they're there and they're devouring up the exudates. Now, a lot of these are going to get eaten by nematodes and protozoa, but this is before that has occurred. And what happens is they form a bacterial slime. When you have a bunch of bacteria, they form a slime in order to be able to hide from their predators and in order to be able to trap food sources. And so that's why you brush your teeth in the morning. They have a slime on it. You have to use energy in order to remove that slime, and it's a real protective layer. These certain bacteria, these rhizophagy bacteria, are living in the slime with a bunch of other bacteria, some of which are going to get eaten, and they smell something very familiar. They smell a popcorn smell. And this popcorn smell indicates to them that there is a empty slime facility right next door, and they should go live there because there won't be so much competition. And so they move into that new slime area attracted by this smell. Only if there isn't a slime area, what they end up doing is breaking through these very thin walls at the tip of roots, very, very thin walls, and they break through it thinking they're going into a new slime colony. And they break through these cell walls, and they end up in a particular spot in the cell that's known as the periplasmic space. Now, I'm throwing a lot of science at you, so again, I try to make it easy. The periplasmic space is this little space between, well, let me make it really easy. This is the cell wall, this is the guts of the cell, and there's this little area around here. The easiest thing to think of is tofu. This is the cell wall, this is the guts of the cell, and there's the water all the way around there. That's the periplasmic space. And these bacteria back through, they smell that popcorn smell. They're attracted again by the plant itself, just like the mycorrhizal fungi are, and they back into this periplasmic space where they find themselves. And in the periplasmic space, they live and they multiply, because every 20 minutes they multiply. So after 20 minutes of being in there, they're doubling their numbers, and they're cycled. There's a little cyclosis going around inside the cell that moves stuff around, and this moves as well. And so here you can see, here's another picture, different kind of bacteria, different kind of plant cell, but these are those endophytic rhizophagy bacteria. All right, so they're inside there, and when they get in there, the plant goes, I got to be careful. I don't want these guys to take over and kill me. And so the plant sprays the bacteria with something known as a ROS, a reactive oxygen series. ROS is quite simply what causes sunburn. When you get sunburned, it's because your cells are producing a ROS to protect themselves from the UV rays. Well, they produce a ROS to protect themselves from these bacteria. And in fact, I even found a ROS with a sunburn just so that we could tie everything together. All right. Anyway, this ROS has the effect of stripping off the bacteria's cell wall. And so the bacteria end up without cell walls, and the plant cell absorbs the nutrients that were the bacterial cell wall and use them to grow, which is a pretty amazing thing. Now, the bacteria, as you can imagine, they don't like to get sprayed. And it does a couple of things. It stresses them out. But by the same token, I should add, it also stresses out the plant. And if the plant's not careful and sprays the wrong or the too high a concentration of ROS, it hurts itself. And so by spraying the ROS on the bacteria, it has to strengthen its own cell. And so the plant becomes stronger as a result of these bacteria. And so they get this induced stress resistance, which lives throughout the plant for the rest of the plant's life, that makes the plant stronger. Now, it also turns out that the bacteria don't like it. And so they do two things. The first thing they do is they produce ethylene. Ethylene causes the plant cell, it's a phytohormone, and it causes the plant cell to stretch. And then they also produce nitrate. So the ethylene and the nitrate react and weaken the ROS. They produce different things that weaken the ROS so that even though they've lost their cell walls, they're not going to get killed by continued application. So it weakens the ROS, and these wall-less bacteria continue to cycle in that periplasmic space, and they continue to multiply as they produce this stuff. The cell is stretched, and they take a little bit of carbon, these bacteria, and they form a cycle, which is very, very important. In order for the bacteria to keep alive, and in order for the plant to keep alive, these cycles form, and the cell continues to stretch, the bacteria continue to multiply, and they produce their ethylene. And the protection that they produce is actually nitrous. They take nitrogen from the air. Now they're inside the plant, and they fix that nitrogen to form nitrous oxide, which also weakens the ROS. And so they got this ethylene and this nitrous oxide weakening the ROS. What they're doing is they're fixing nitrogen inside the plant cell, just like rhizobia bacteria do. But this is completely different. This is inside the plant cell. And it turns out that if the rhizosphere cycle is going on, up to 40% of the nitrogen that a plant uses, I'm going to repeat that, 40% of the nitrogen that a plant uses comes from this internal nitrogen fixation, which we had no idea was going on before 2018. So if you haven't ever heard of this before, it's because no one has. In fact, I would say you're probably a group of 4,000 people who may know about rhizophagy, but everybody's going to learn about it. So what you've got, again, this is the depiction. This is the periplasmic space. The bacteria are moving around, and they're being cycled around. And inside from the periplasmic space is a membrane and all the goodies that are inside the chloroplast and all that kind of stuff. So, but we're interested in this area right here. And you continue to get growth as these bacteria are cycled around, multiplying. And again, they're very, very, very small, and they multiply and they begin to form structures like this. This is a multiplied wall-less bacteria, and they're all throughout the thing. And then they begin to form like this. They begin to conglomerate. You get more and more of them because you continue to get this multiplication, and they begin to clog up the pathway, so to speak, and back up against the wall of the cell. And when they back up against the wall of the cell, the ethylene concentrates against that wall and forms a tube. And these are the tubes that are being formed off of the main cell of a root tip. If you look carefully here, you can see this is what the tube. See the tube being formed, and it's there. You see a little bit of the tube here. It's not a brand new cell. It's part of this cell right here. It's this little hair that grows out of the root. Wait a minute. It is. It's a root hair. So here's a shocker, folks. Every single root hair you've ever seen has bacteria in it. Why do I know that? Because the bacteria are what cause root hairs. We were all brought up to believe that root hairs grow so that the plant can get nutrients. They go out into the soil, blah, blah, blah. Well, that's not true. They grow because they're putting these bacteria into them. They're getting bacteria filled. They happen to be very thin-walled, and I did a tremendous amount of study, again, because it was COVID and because I had the ability to be able to do it. These are very thin-wall structures, and they open up. And so there's no wonder that if you read Teeming with Nutrients, one of the other books I wrote, which talks about how plants take nutrients in, the nutrients are able to move through these incredibly thin walls. And so they do take up nutrients, but they are formed by bacteria for a completely different purpose. Because what happens is they continue to multiply as they move into these cells. So they're in the cells and they begin to multiply and they get conglomerate up at the tip. And this blue indicates that there's nitrogen being produced from these little guys. That's the dye. And then all of a sudden, they are blown out of the tip of that growing tube, which we call a root hair. And they go back out into the soil. And here they are conglomerating in the tip, and boom, out they go. And here they are, blown out into the soil. These are wall-less bacteria. So are these. And they grow back their cell wall. And in 2 or 3 days, they move back into a new root cell and form new root hairs. And it doesn't happen just once. As that root hair grows, you get explosion after explosion after explosion, and then they go back into the soil and they grow back their cell wall. This is completely amazing stuff to most people. They work, they eat, they travel, they repeat, they go back. It's an incredible system. It's now called the rhizophagy cycle. It has been observed, obviously, in many different ways. If you remove the bacteria, you do not get root hairs. Wow, unbelievable. So what we used to think is that the plant, by producing the exudates and putting the exudates out into the soil, was sort of farming, and then it would gather in the fall. It would bring in all the nutrients, and that was it. But what we're learning is they also are herding. So they're ranchers as well as farmers, and they go out and they bring these bacteria in. They shear off the walls. They eat an occasional leg of lamb, and then they put them back out into the paddock to grow back the wool again. It's absolutely an unbelievable thing. It's known as rhizophagy. You may not have ever heard of it. You will hear lots about it because it turns out that it's a very useful research tool and potentially an unbelievable substitute for actual chemical fertilizers. So that's what the world's been looking for a long time. Now, those rhizophagy bacteria that move in are endophytic bacteria. And as these roots begin to grow, you end up with little cracks in the main root itself. And because bacteria are everywhere, and because bacteria are always looking for an advantage over other bacteria so that they can go to a place where they can breed without predators and where they can eat without competition, there is a bunch of bacteria that would have been eaten perhaps by nematodes and protozoa that sneak in the cracks that form when these root hairs form as well. And so they sneak in, they're very, very small, and then they're capable of moving throughout the plant in the phylum or the xylem, and even in the athoplastic pathways in between cells. And so you end up with endophytic bacteria throughout the root, and they move into the plant itself. Some move in through stomata, but most of them come in through the root system. And some of them end up in the flower. And as the flower is forming seeds, they get trapped in the seed. And they're inside the seed so that when the seed is planted in new soil, the seed brings with it the kinds of bacteria that it needs in order to grow properly. Again, an unbelievable discovery. There are corn plants, species that are 400 years old, and if you plant one today, it has the same bacterial mix or very close bacterial mix to what it had 400 years ago because it carries its seeds. This is probably where terroir comes from. Not the soil, but the microbes that are in the plant that come from the soil and get passed on. It's an amazing thing while we're studying. And so you get the bacteria, they get trapped, When the plant germinates, the bacteria move into the soil. As some of them end up as rhizophagy bacteria, some of them end up as endophytic bacteria, and it's a beautiful, beautiful system. These bacteria, these endophytic bacteria, are capable of producing hormones. And so they produce the key hormones that you need for plant growth. Now, the plants obviously can produce these as well, but when they get assisted by these bacteria, it's a wonderful thing. And why would a bacteria do this? It would produce these things inside a plant because it's tricking the plant into allowing it to live inside the plant. So the plant says, oh, yeah, I get some oxygen out of this. Well, gee, I get some ethylene out of this. And I'll let these guys stay here. And the bacteria gets to live without the competition. It gets a source of food without competition, and everybody lives happily ever after. So you end up with all sorts of things as a result of these bacteria. The stress that's created it causes some wonderful things to happen. This induces systemic resistance, but you also get all of these things occurring as well as a result of these bacteria moving in. And I might add some fungus, particularly yeasts, do the same thing. They're not mycorrhizal. They act like these bacteria, and they go through an endophytic phase of their life. And these endophytic bacteria help plants to deal with abiotic stress. They can lower the freezing point of water, or raise the freezing point of water, excuse me. They can help a plant deal with salt, with flooding, with drought. They can help plants deal with pathogens that are attacking them. The plant can selectively allow in a bacteria that's able to take out some of its pathogens. It's a wonderful, terrific defense system. If you can get the bacteria to do your fighting for you, all you give up is a little bit of space and maybe a little bit of carbon. Some of these bacteria are capable of doing things like chelating iron. And so they'll enter into a plant root, they'll live inside the plant. And when the plant is either got too much iron or doesn't have enough iron, they can hop out into the soil where they can gather up all the iron together, or arsenic together, so that it doesn't go in and poison the plant. So they do all sorts of wonderful things. There are, Tad and I were talking, just the beginnings of endophytic bacteria that are producible and are available. These top ones up here are things that you can get at cannabis grow stores. They're beginning to use them. This one fixes nitrogen. There's different kinds depending on what areas you live in. There's one from Brazil and Russia, et cetera, et cetera. This one is one that people are beginning to look at. These can be stored relatively easy. This requires freeze drying. So people are trying to figure out how to use these things. But what big industry has discovered is that it's possible to find and to isolate endophytic bacteria that do things to your plant that you need them to do. So, for example, if you're growing a tomato and you end up with powdery mildew all the time on that, if research has it proper, they're going to find the right bacterium to put into the soil so that it goes into the plant and fights off that powdery mildew, so that you don't end up getting that problem. They're not doing it for me and you, they're doing it for the big guys, but it'll come down to our level, no question about it. And these three are already on or getting close to our level, just as rhizobia bacteria have come down to our level. You can find rhizobia bacteria, nitrogen-fixing bacteria, wherever you buy legumes as a inoculant for your peas and other legumes. So the really obvious takeaway: plants really are in control. Whether you go by the original soil food web, or you move all the way through to the modern view of the soil food web, which includes the rhizophagy, they're in control. And we like to think we're in control, but when we think that, we end up taking actions that are not necessarily in the soil food web's best interest. We interfere with the way the soil food web works, and we end up having to do a lot of work ourselves as a result of taking away the ability of these invisible microbes to do the work for us. So plants are definitely in control, and they're amazing, which is really the most amazing takeaway. They do things we just completely had no idea that they were doing. We keep getting better and better at microscopy and and staining, and as a result, we're beginning to see what plants can actually do. This is where I usually pop in and say, we've got to stop teaching our kids about dinosaurs. Useless. We need to start teaching our kids about microbes because they impact everything we do. And they're beautiful. They're absolutely stunning and gorgeous, and we can make them every bit as interesting as, as the Tyrannosaurus Rex and all that other kind of stuff. It's amazing to me that a 4 or 5-year-old kid knows all the dinosaurs, knows even how to spell some of these incredibly complicated words I can't even say, and yet has no idea what a bacteria is. Chemicals obviously have a negative effect on endophytic bacteria. They throw things out of balance, so you got to be very careful how you use it. You got to use the best practices possible. All of the things that keep a soil food web alive also help the rhizosphere cycle and endophytic bacteria. Mulches, leaving leaves alone, using compost, even compost teas, compost extracts. These are all things that help the soil food web, are considered to be best practices. No rototilling, which breaks everything up and destroys the soil food web, or at least impacts the soil food web so that it has to rebuild itself and you lose that time. So all of these things are important. You have to feed the soil. The soil is everything. You can test things. My favorite test these days is something called a microbiometer. Yes, you need to have an NPK test every now and then so you know what your soils have in it and what they're capable of doing. But you need to know whether what you're doing is helping the soil food web or whether it's hurting it. Go look at www.microbiometer.com. It's this really cool test. You use your cell phone and you can tell whether you're increasing or decreasing the biomass, the bacterial and fungal biomass. If it increases, you're doing good. And if it decreases as a result of what you're doing something wrong. And don't disturb the soil. Prevent compaction, no tilling. Be careful. Your soil is the most valuable thing we have. Don't sterilize seeds. That's a silly thing to do. And don't necessarily use sterile soil either. Those seeds contain the bacteria that your plant needs in order to live. When they germinate, you want to have them be able to do so. Use mulches. They provide organic material, which all of the soil food web needs. Don't rototill, don't use soil, reuse it. It contains exudates, it contains bacteria, it contains organic material, and compost, beautiful stuff. Use it all the time. Make compost out of the same plants you're growing. It contains a lot of the goodies that your plants used and would use again. Test your compost. Some plants, because compost has different kinds of bacteria in it, like vermicompost, and some plants like thermal compost more. Which ones? I don't know. We got to do the research, and this is a home scientific experiment. But you're going to find that your broccoli is going to do better in one kind of compost versus the other because the bacterial composition is different, and you'll get different endophytic bacteria along with the ones that come along with the seed that you're planting. So a not so obvious takeaways. You got to read these books, folks, not because I wrote them, but because this is the science behind organic gardening. And if we've learned anything, it's that we need to be organic gardeners. We have way too much plastic in our lives. We have way too much plastic in our soils, for example. We don't need to add chemicals. We just have screwed things up so badly. We know it. We know it's caused bad things. Start with teaming with microbes, move to teaming with bacteria. learn how, and then go to endophytic fungi, and then learn how the plants use all of these things to grow. If you do these things, you will be a much better gardener. Chemical gardeners are not good gardeners. To learn how to use the soil food web, on the other hand, is how you become the very best gardener you possibly can be. And I say, is this the end? Probably not. There is no question. Some other thing is going to be discovered. Who knew in 2017 that you could get 40% of your plant's nitrogen just by simply making sure that the rhizosphere cycle is going on? No one. And so there's got to be more stuff out there. There are people looking for this stuff all the time. If you're taking notes, write down the name of Dr. Elaine Ingham. She is the guru of the soil food web. Write down the name of Dr. James White. He is the gentleman at Rutgers University who has really refined the rhizosphere cycle. And that's it, folks. I appreciate your attention. Be happy to answer any questions you have. Thank you. I can't hear you, but I see you're clapping.
Oh.
Any questions? I can hear you.
Oh, okay. Were there any questions I can relay or? Oh, one.
How did you find out about plant a row for the hungry?
Oh, how do you find out about plant a row for the hungry?
Oh, I'm glad you asked that. Plant a Row for Hungry is sort of a self-service program that we used to be the Garden Writers of America. We're now the Garden Communicators International. And if you go to gardencommunicatorsinternational.com, you'll find a page that talks about Plant a Row for the Hungry. All you have to do is share food with somebody else. As a club, for example, just to encourage everybody to plant one row in their garden, the gardener takes that food either to a neighbor that needs it, to a church or a synagogue, to a food bank. There are places where you can take food to a prison. The prison gathers the food. We've got hardware stores that gather food. We've got nurseries, garden nurseries where you can bring in excess food. They'll take it to the food bank. There's no rules to this other than you plant the row, you distribute that row somehow, and nothing slips between the lip and the cup. There's no money involved. There's no administrative services involved. This is a people-to-people campaign, and it is unbelievable if you just Google Plant a Row for the Hungry, how many different programs people have come up with. We do it in Canada now as well. They're beginning to do it in England. 35 million people go to bed in North America hungry every single night, or at least not knowing where their next meal is coming. This is such an easy thing because gardeners share, and because most of us tend to just sort of leave stuff in the garden, when the snow comes for the moose to eat. And that's a stupid thing to do.
Yeah.
Could you expand on the value of cover crops that are nitrogen fixers and how they actually make the nitrogen available?
Are you able to hear that, Jeff?
Not really. Talk about nitrogen fixation crops.
Can you expand on cover crops and how they make
cover crops? Sure. Yeah. Well, cover crops are mostly legumes, and they produce the nodules that you're familiar with, and inside those nodules they produce nitrogen. A little teeny bit of that nitrogen leaks out into the soil and is used by whatever plants are growing there. But what happens is the cover crop dies, and when it dies, the nitrogen that's in the cover crop itself, in particular in the nodules, goes into the soil and is then usable. That's why you can plant a cover crop in the fall. It's producing those nodules. It's producing nitrogen for itself, and then in the spring, you tamp it over, or you just rake it over, maybe a little light tilling, and that moves that material into the soil the dead plant then provides that nitrogen, releases it.
Clovers are the most common one you hear of too. Just
Right. And by the way, I've been testing a new clover. It's called micro clover. It's not really new, but the new trend is for people to put in not lawns but meadows, to turn their lawns into meadows. And one of the easiest ways to do that is to add tremendous amounts of clover. This micro clover it only blooms once a year. It's beautiful stuff. It's worth taking a look at. But it pumps in nitrogen like crazy. And in addition to using it on my lawn, I've used it on my tomato plants. And I gotta tell you, it makes the most gorgeous tomato plants I've ever seen. This mound of beautiful clover. And then after I harvest the tomatoes, I'm going to kill it. So next year they're going to get the benefit of this nitrogen.
How do you suggest using compost if we're not tilling, just as a top dress?
Yes. The compost, and you really don't need very much. 1/8 of an inch is enough. An inch is, wow, a tremendous amount. If you put it on top of the soil, the microbes in it, as well as the rain, etc., work the microbes down into the soil, down into the growing zone in a couple of 2 or 3, 4 or 5 months. So yeah, just put it right on the surface. It has in places like Anchorage, and you get a little bit of cold weather. It acts as a little bit of an insulation layer. We like to put the compost down and then put leaves on top of it as mulch, so that it holds the temperature in the soil to 29 to 30 degrees in our frost zone. It raises the frost zone so that we don't get quite as deep a frost, etc. I love compost. It's great stuff. And Tad makes some very good compost, by the way.
If I can follow up on that, I've also read studies though that are showing that we're getting too much organic material in a lot of our garden soils because of all the compost we have.
Yeah, I've read some of those too. I'm not. It's hard for me to. Well, let me put it this way: most soils, most regular soils, has about 1 to 2% organic matter. Really good organic garden bed might have up to 5% organic matter. Can you have too much organic matter? Yeah, I suppose you could if you weren't getting soil structure. If it was replacing the individual, but it's hard for me to see that. I mean, I can see how maybe if you use manure in your compost, you may end up with too much phosphorus, or too much potassium. That would be probably a more thing. But yeah, I don't buy into it, frankly.
Thank you.
And that doesn't mean I'm right, but I just don't buy into it. I put as much compost down as I've grown in directly in compost. I organic matter all the time. I never rake a leaf unless it's for putting down on the garden. I grow potatoes in just leaves, which is all organic matter. They do just fine. So yeah, I have a hard time. But again, these are scientific papers. People, Linda Chalker Scott, etc. They write these things, and there's got to be some validity to it. But I haven't had that problem.
yeah.
I saw a picture of red worms, and I've never fully used to advantage the castings that I generated in my worm bin. What's a good way to distribute that stuff? And what is the value of the worm casting as far as who's living in it?
Can you hear that?
Yeah.
What's
So in
the-
other words, how do you collect the worm castings, and what do you use them on?
No,
How do I best use them? I've got masses of it.
she's got a lot, but how do you best utilize them, and what's the value of them?
Right. Well, I use them in 2 different ways. We also keep worms. In fact, now that we see the snow, I'm gonna have to move it inside. You can use them in by putting them in liquid, and sort of making a worm compost extract and using that liquid on plants. I don't know really why I do that, but sometimes I do. Otherwise, I put it right on my plants. Now you got to be careful because they will contain, little worm casts, little eggs. And so you got to make sure you've got the right worms. You don't want that new worm that's moving in that does the fast squiggly wiggly. I'm not a particularly big fan of adding night crawlers if you don't already have night crawlers in there. The red wigglers seem to be safe, but I wonder whether we're really doing the right thing by using them. But using the castings themselves does make a lot of sense. They contain a concentration of the nutrients that is higher than what went into the worms. So I don't remember exactly what the numbers are. It's X amount of times more phosphorus, X amount of times more nitrogen, it's got higher concentrations of the nutrients that you want for your plants. And that's why it's so good.
There's research on adding worm castings to potting soils, for example, and they found up to I think 15% by volume. And again, this depends on the quality of castings and things like that, but they actually got increased biomass production, growth, disease protection. So the worm castings have a lot of benefits in soils in general.
Yeah. And I would think because they contain so many nutrients, they raise the bricks of the plant. And if the bricks of the plant is over 9 or 10, you don't get insects anymore. So yeah, it's great stuff. It's every bit as good as compost. It's just one's thermal and one's biological. So there's a different mix of bacteria in them.
Any other questions? Okay. All right. Well, thanks, Jeff. We will let you go then, but I really appreciate.
Pleasure. My pleasure. If anybody has any questions, let me know. And I always tell people, if you buy teaming with microbes, and now I'm going to add in teaming with bacteria, and you don't like it, I'll buy it back at 3 times the price, because I think it's that important that you read it. And if you want to read it in French or German or Slovakian or Turkish, Portuguese or Spanish, we can arrange to get you that copy. So.
And Audible now, huh?
Oh yeah, it's on Audible. It's on discs. It's the same person that read John Stewart's book reads mine.
Oh, cool. All right.
Thank you very much, folks. What a great audience. Thank you.
Thank you.